So we can start with the discussion now, and please raise your hand if you have any questions or any comments to make. Nobody wants to start? Okay.
Thank you for the chance to be here and listening to your presentations. All of that I found very interesting. Just a single point. I have several points, but some of these will come up later on, I suppose. But in principle, a three-way comparison seems sensible. However, in a clinical development program, I don't know if there's any statistician here. If you make more than one comparison, people jump up and say, oh, that introduces multiplicity issues. Because if you make several comparisons, you can come up with several different results, which then have to be put together sensibly. Has anyone got any comment on that in the panel?
I'm not sure I can answer your question. I don't know whether we had given scientific advice on this. I see some people from the Scientific Advice Working Party in the room. They have to ask for multiplicity. This is indeed something to consider. Yes, Andrea Laszlo?
I'm Andrea Laszlo, CHMP member and scientific advice. I would not remember that we have given clear advice on this specific topic. However, as was said, I believe that some adjustment for multiplicity might be necessary. In that case, choice of sample size would need to be made accordingly.
Just continue. In terms of quality assessment, would the quality assessors be using some statistical principles here as well? But quite often these days, that comes up as well in terms of using statistics for quality comparison.
I think we just had a very interesting discussion on this topic, and I think the conclusion is that this is not ready for prime time yet, that we could get some valuable supportive data from statistics. But this is something new, and we need further discussions on this. And the feeling of the group was that this is more, yes, nice to have and could be further developed and could become important in the future, but not at the current time. So I don't know whether anybody wants to add to this. Andrea?
Well, if I could just add to this, I would expect that within the upcoming scientific advices in the near future, we may actually come across one of such questions. And then that's the first point, of course, when we are going to address this and discuss in depth from the statistical point of view. However, until now, we haven't actually had such questions within the different advice requests that we have worked on. So apparently this is, as said, indeed some topic that is coming up new onto the table.
So there was another question back.
Yeah, my name is Thilo Netzer from Pharmalex. I think before embarking into such a discussion, it would be interesting for me to understand whether when we are talking about a three-way comparability exercise, this means that you really do this side by side. Because you could also do it in separate trials and then put together the data and integrate it. And this would alleviate, the issues with respect to statistics. So my question basically is the expectation here that this is really done with respect to quality and clinical aspects in a side-by-side studies, or could this also be achieved by separate studies really comparing, let's say, only two out of these three combinations?
I think it is much preferable to have it in a three-way comparison, really. That's the best comparison you could do. It's always much more difficult to make an indirect comparison. That's the same would apply to generics, bioequivalent studies. You couldn't do them this way. So it really is preferable to have a three-way comparison in the same study if you need to do that study. I don't know whether we have any.
I'm Josie Dratwell, and I represent patients with primary immunodeficiency. The last speaker spoke about the fact that only part of the EU patients have access to biosimilar because of the cost. So my question, obviously, is, do you see that changing, or where is your market?
Okay, if we have a look, for instance, about what happened with filgrastims in the UK, and there we have clear precedents, there with the introduction of biosimilar filgrastims, you realize that there was a significant decrease in price and a very significant increase in patient access to this treatment. This is the precedents which we have for the time being, which I remember, but I'm strongly convinced that this will happen also with the other drugs. And if we look on modern antibodies, the patient access is somewhere in the, let's say, low double-digit % figure. And we think that there are much more need for these products in Europe.
Thank you. I just saw a request from participants via Adobe Connect. Everybody who speaks, please identify yourself briefly. State your name. Thank you. And I have a question here, or a comment. So Mr. or Mrs. Tota says, multiple comparisons with cohort studies are justified by two-way ANOVA analysis. Welcome expert comment on this. I think we are not talking about cohort studies here. We are talking specifically about PK and maybe PD studies, not efficacy trials or cohort studies. But anybody wants to comment on this? We have also another question or a comment. A three-way comparison in the same study would need a substantially higher number of patients, which could cause difficulties in recruitment. I believe this also refers to really large clinical trials. That's my impression here. But does anybody want to comment on this? Please here.
So this is Sigrid Balzer from Sandoz. I mean, the comment that I would make is, I mean, usually if you have to do a three-way comparison, a parallel group design, that certainly increases then the sample size which is needed for such a study. But also what I would like to keep in mind is, I mean, there are compounds where you could do a single crossover study, which is more efficient than doing this setup in a parallel group design. And I think for those, it may make sense to do two separate crossover studies and then do a cross comparison between the two reference products. I mean, that could be much more efficient.
Thank you very much. Are there any further questions? Maybe I can, oh, yes, please.
Cornelia from Merck. I think I really appreciate that there is a possibility now to really make a case-by-case decision on whether a three-way PK study is needed at all. And I think here the fact that analytical development has evolved so much over the last 20 years, we know much more about the quality of the products, of the reference product, as well as of the biosimilar products than it was at the time when the originator products have been put on the market. So I think here, if you really have strong data between reference products sold globally, knowing that these products are approved on the market and patients treated with that, then I really appreciate the openness to look at the data and then decide whether really another three-way PK study is required at all. Because as stated here, depending on what you're doing, whether you do a parallel study or a crossover study, that will definitely either enhance the numbers of subjects tested or the time to carry out the study. And from a scientific principle, it may not be needed. I understand that at the moment there is too few knowledge because not a lot of companies have performed those studies. But I think as we are talking about a guideline, leaving the door open, that once it has been demonstrated by a lot of companies on different products, that indeed when you show that quality-wise you are within the ranges, that in future we can defer from doing three-way studies.
Yes, thank you. I would like to ask a question to Dr. Koretsch regarding this requirement for PK/PD studies. We should also consider that the formulation, of course, is important and could have an effect on absorption of the molecule. It's not only the active ingredient itself. So, but I would like to ask Dr. Koretsch, why do you think we should always have a PK/PD study?
Oh, I think
or is it a case-by-case decision?
I think I did not express myself correctly. I said in many cases. Definitively, I think we are all around the same thing, that it cannot be a systematic position to say it will always be needed or it will never be needed. Of course, it will be a case-by-case basis. Our only point was to say that, again, the biosimilar maker does not have access to the full package of data of the innovator. That's fact. And this may, in several cases, in many cases, maybe lead to the need for a human PK and PD study. But our position is not to say that this would be systematically requested.
But you agree that the biosimilar manufacturer would have to fully analyze also the reference product from the other jurisdiction. So,
yes, the same
and
way,
do a full
absolutely,
side-by-side comparison,
yes.
And therefore would know what's really in that product.
I mean, basically, I'm just heard the same thing from all the parties. I would say the steps are the same. Step one, analytical, in depth, and then based on this data and the remaining uncertainty, the need or not the need for a clinical trial. I mean, for, as you say, for a human PK trial. That's I guess that this is the basic position of everybody. The only thing, again, here that we are bringing the fact that all the data for the originator are not available to the biosimilar maker, and this may impact the decision to determine if a study is needed or not needed.
So we have a further question?
Siegfried Schwarze, European AIDS Treatment Group. I'm sorry if I have a very basic question. What defines the similarity of a drug and a biosimilar? Is it only the pharmacologic properties of the drug, or is it also the biologic action of the drug? For example, interferon alpha 2A and interferon alpha 2B, would they be considered similar?
No, they wouldn't, because they have different amino acids sequence, and therefore they wouldn't be. So that's one of the mainstays, I think, that has crystallized really over time, that we definitely want to have the same amino acid sequence.
Okay, so it's about the drug itself and not the biological action, right?
The action is, of course, very important, so they should have also the same pharmacological action. So the molecule itself should be as close as possible to the reference product. So it should be engineered in such a way that it really resembles in its physical-chemical
characteristics. Okay, but where are the limits, you know? You say as close as possible, but there must be defined limits.
It's a matter of assessment, really, in the end. A matter of assessment and experience. And I do think that we do have experience now with assessments of such applications. And usually we have a lot of questions on the comparability exercise that has been performed by the applicant. And I see that we have some biotech people also from the regulatory side in the room, and maybe they want to further embark on this. But this is in the end really you can't provide clear limits, because you have different active substances, and you have different impurities and so on and so on. It's very difficult. You cannot provide one margin that would fit everything. That's not possible. It's a case-by-case decision.
Okay, thank you very much.
Yes, please.
Keith Watson, RXL Consulting. This idea that a case-by-case basis could apply for three-way PK studies, how would the CHMP provide definitive advice to a company where, by assessing their quality data to make that decision? Because my understanding is that the clinical trial applications don't really have the timelines in order to do a thorough assessment and make that decision, neither the scientific advice. So how would you be able to make a judgment to a company to say, yes, your quality data is sufficient, such a three-way PK study isn't required?
I think that's very difficult. Indeed, in the scientific advice, we do not make a pre-assessment of the data. That's not real. It's more the strategy of your development plan. It's not the same way FDA might do it. And so this is clearly different. And I think this is some risk the companies have to take. I guess of course, I think the assessors that look at the scientific advice, at the briefing documents, they will look at the data, but they might not be able to look that closely or in depth at the data and provide really the final decision. I think this is the company themselves that have to, in the end, have to make a decision whether or not they go forward without a three-way PK comparison, for example. But we realize that there is really a lot of experience also within the companies. Yes, please.
Thank you. I wanted to switch topics a little. This is Richard Marcus from Amgen, and this personal perspective or point of view on the new potential definition of a biosimilar that's been posed, even though as was presented, there exist multiple definitions in different areas. But I don't believe a version, using that terminology of a version, is really appropriate and potentially misleading. I think when we talk about a version, that's usually referred to as a change within a single manufacturer, and take it even out of pharmaceuticals. For example, take if Google were to prepare or offer a spreadsheet application, it would never be considered a version of a Microsoft Excel, even if it had the same features and attributes of Microsoft Excel. It's a unique product. So I think we are blurring the lines when we use the word version. And I think we should keep it clear that a different manufacturer making a biosimilar is still making a different product. It's highly similar to the other product, but it's still a different product. It'll have its own life. It can have its own process changes post-approval. So I think that the terminology is potentially misleading in that way. So I'd recommend sticking with the prior definitions of it's a biologic that's highly similar to an originator product or something on that order. Thank you.
Okay, thank you for this comment. I'm sure we will further discuss this within the group. Yes, please.
So I'm Åsa Remrat from Novo Nordisk. Coming on to that, at the end of the day, it is actually a product we're comparing to, especially in the clinical program. So I would also say that a biosimilar, at the end of the day, is highly similar to a product, not just to an active substance, which could have a different administration route, for example.
Maybe you want to... Oh, sorry, Martin?
Thank you, Martijn van der Plas from the Dutch agency. I was probing a little bit along this line. Would it be helpful if there were more separation between the product, the drug product, and the active substance, in which then could argue that the drug product could be similar because it will always be a unique product because two different companies will make two different products, but that, however, the active substance, the active ingredient of the product is similar or highly similar or the same or whatever terminology you might want to use, but that at least it's clear that these are two different aspects with two different but overlapping requirements. Would that be helpful?
So I'm not sure if that was a direct comment to my question or comment, but I think it's helpful to have distinction between between substance and product for sure, and that can be a theme in general. I think having matching, quote unquote, matching quality attributes within the range of what is observed in a drug product, for example, when it's reverse engineered, is still what's needed and appropriate to be considered biosimilar, but it still does not make it a version of the reference product. It's still a unique substance and a unique product. I would contend that when there are multiple manufacturers, each developing a product, a biosimilar product, they will all still have unique features within their active substance, for example, that would uniquely identify who the manufacturer is when you just look at the list of quality attributes. So they will all still be unique to themselves by manufacturer and not blur those lines.
I saw Jörg Windisch.
I think this all goes back to the discussion we've been having about identity or identicality that's been out there for a long time, where it's simply a matter of fact that if you look at a glycoprotein, you're not looking at a single substance. You are looking at a defined mixture of closely related substances, which varies in any biologic from batch to batch and somewhat more pronouncedly after manufacturing changes. So that's also true for an original product, also true for a biosimilar, and the goal, of course, is to lie within that variability of the original product. So to say that, one thing is one thing and it's always the same thing and only that can have that name is simply not appropriate. So I actually thought that using the terms known and using the word version was actually a good way of addressing the issue and still saying we're talking about biosimilars and that identity or identicality is not really a principle that applies to biologics in general.
Yes, the next question,
I think Jörg made the point, because I think whatever rule, my name is Sumant Ramachandra from Hospira, and even of the products that we have developed, and we have 3 biosimilars in Europe at this point, whatever rules come out, I think when we talk about comparison of the products, that version language is actually a very good one, because even within the originator product, the changes that have been shown to be made in manufacturing processes have resulted in different isoforms of those particular products. And actually, if you use that particular terminology, there was clinical comparability shown, but the actual product attributes in terms of the characteristics of products actually changed. So we have to be very, very careful that the rules that we are starting to put around biosimilars actually apply to biologics in general. And you're going to see these lines blur over time. And so when we look at product attributes and quality, they have to be for the biologics field rather than for the biosimilars field over time. Because the products will change over time, whether it's a biosimilar or an original biologic.
Thank you. We don't have another question.
This is Mark McCamish from Novartis Sandoz as well, and I would agree that the name version is really a very nice name that's been thought through, and the question is one of communication, and I think we've all run into this when we're talking about biosimilars, particularly to clinician, and the guidelines as they go out are based on communicating to a broad population, and all of us around here are really experts in this area, and so we can in fact have debates on small differences, changes, et cetera. But when you get out into the field and you're talking to the end user, physicians, healthcare providers, it's really important to have a common term that can be used. And biosimilar itself is challenging as you begin to communicate this. So the version terminology, I think, is very apropos in terms of communicating what we're trying to do, create the same substance, same within the terminology in terms of clinically relevant that's there. Again, it's the same terminology that has been used for comparability and extrapolated here from a scientific perspective. And how you pick a word that communicates that to a broad population is indeed challenging. And I think the terminology that has been proposed by EMA in this guideline is a very nice terminology that allows that communication for individuals who will be using this.
As I said before, very difficult to make everybody happy.
Trying to get this, I don't know what I touched.
Narayanan?
Thank you. I'm Narayan from Biological Advice. I'm a little bit perplexed as to why the word similar doesn't appear in the definition. Was it intentionally left out? Or is it not simpler to say it is a similar version of the originator product?
But you pressed that because as soon as I disconnect, then it will show that I'm not there.
I think
You're online there?
Yes.
Okay, that's
I was
what I...
the rapporteur for this guideline.
That's right?
I think we wanted to make clear that this is a
Yeah.
very
Now if you press this, you
close
can
to
get
the active is very close to
Press this on your phone, 65, okay. You're going to
and
disconnect
in a second paragraph
653.
and how similarity should be demonstrated to separate these two issues and not to intermingle it too much, to make it clearer. I think that was the intention.
Yeah.
Jörg Windisch?
Yeah, this is exactly the point. I mean, there's a sentence that comes immediately thereafter that says that a biosimilar demonstrates similarity to the reference product, which is still part of that definition paragraph. So I think it's all there.
I have a request from participants connected via Adobe to whether the speakers could summarize the comments and discussions. And I think that's might be a bit difficult to do, and I would suggest that we do that for written questions. What do you think? That we could do this for written questions? It's also recorded. Sorry? It's also recorded. Yes, in addition, I think I highlighted in the beginning that the whole meeting will be recorded, so I think that's another way of just tracking all the discussion. That actually might be the easiest way. We do have a comment saying that the three-way comparison is clearly the most important topic here and is very complicated. I don't know how the other participants feel about this, whether it's so complicated. Yes, please.
Just on the definition, I think it's Alexander Menzig from Amgen Europa Bio. I think it's important to go back to the directive which says a biosimilar is a product which does not meet the condition in the definition of a generic. So it would be good to have a clarification about why they don't meet the condition in the definition of a generic.
I think the directive says exactly where a biological medicinal product, which is similar to a reference biological product, does not meet the condition. Then you should have more data. I think that the emphasis a little bit different in the legislation. And the legislation also says that the type and quantity of supplementary data to be provided must comply with the relevant criteria stated in Annex 1 and the related detailed guidelines. And then the Annex 1 also puts the emphasis on the guidelines. I think very much emphasis is put on the guidelines. And I think this is a good thing. And I think it's similar in the FDA legislation, because realizing that biologicals are so diverse, you need guidance for maybe different. We do have product-related guidance also, and this is very, very important because they are so diverse, and you cannot put everything in the legislation. The requirements, the same requirements for all of them.
Fully agree.
We have another question from the Adobe audience asking, how are biobetters defined as per EMA or EGA guidelines? And biobetters is, we think the biobetters are products, usually a follow-on products, not biosimilars. And biobetters have, for example, have intended changes made to the molecule to make them better in some respect, better efficacy, which would then imply a different posology or a different safety profile with intended changes to the molecule. So that those would not qualify as biosimilars. Although, as said before, if we do have a biosimilar that is purer, has a better impurity profile, or is less immunogenic, then this would not preclude the conclusion of biosimilarity. We have another question. The question coming in about combining the data with multiple comparators from different regions in the same clinical study. It was raised in the Innovator Industry presentation. Well, as far as I know, we have said in the scientific advices that this would be
I'll
possible
come in 2 minutes.
if really a conclusive, stable bridge has been established between these reference products. And we believe that these reference products that we are talking about are usually more or less the same, from the same marketing authorization holder, and that these reference products usually will not differ a lot. But that has to be clearly established by side to side, head-to-head comparison in the bioanalytical and functional comparison. So that has to be clearly. So I think it's very unlikely that we will have completely different reference products from different marketing authorization holders in the same trial. I think that's not a likely scenario. Thank you. More questions to come. Again, a request. Please state your name before you speak. So my name is Martina Weise for the audience participating via Adobe Connect. So we have another question, please. Please state your name.
Good morning. My name is Laura. I'm from Brazil, from the National Regulatory Authority. And that biobetters should not be considered biosimilars, I agree. But and change to improve efficacy will not lead us to a biosimilar. But we have a specific case in Brazil that a company wants to develop a biosimilar of bevacizumab and wants to include another clinical indication. In this case, the approach could be, I think this case is not covered by the guideline, and I don't know if the approach for that case will be a new product development plan, or what will happen, because I don't know if the scientific advice committee have that example, but in our country, a company wants to include the macular degeneration indication for this molecule and submit as a biosimilar. I would like to know.
But the molecule is biosimilar. You mean
Yes.
they are developing it as a biosimilar,
Yes,
but just want to include that indication.
Yes,
That's
they
a difficult question, I guess.
They will do another clinic. They will do a
They will do an
clinical
extra
trial.
clinical
Yes,
trial.
yes.
So, in fact, I think if they have a known development program for that indication, that is more a standalone than that.
Yes.
But that would not be a biosimilar application then. It will, because in a biosimilar application, at least here, I don't know your legislation.
On the right-hand side.
You can only claim the indications of the reference product and not more. And then the question is, what could you do afterwards? You could maybe apply for a new indication once you are licensed, but based on own data.
Mhm.
But we haven't had that scenario yet, I have to say. I can't really, I'm not sure about the legislation. Andrea, Laszlo?
Participants' microphones have been turned off.
Right. This is Andrea Laszlo, and in extent of what you said, Martina, we have had discussions previously whether an unauthorized indication could be used in a clinical comparability study, and we have agreed to that. But this would not automatically imply then that there is an indication for such, given in 4.1 for the biosimilar upon authorization. So if then someone would want to include a new indication, I believe that would need to be based on a standalone development.
Yes, please.
Cornelia Un from Merck Serono. So do I understand correctly that for a product, if it's scientifically justified, a non-approved indication could be used to demonstrate efficacy? And I think that's fully in line, which has been discussed and communicated by EMA well before. Now coming back to the point, what Laura has raised, I think I'm also perfectly aligned that when you have a biosimilar application, you only get the indication which has been approved by the originator. However, as the efficacy has been demonstrated in one trial in that indication, I could see it if you do a second trial in the new indication and file afterwards a variation where you have two independent trials. I wouldn't see why that should not be possible, even if the product has originally been licensed as a biosimilar, because for the new indication, you would have still two studies to get that approved. And I think that could be also a scenario to be thought of in the European Union as well, because we know all about the product and we know why this indication is not approved in Europe.
I think that's a very difficult point, and it wouldn't be really easy, or I couldn't see how this could work. You also should realize that the products are different, standalone products on the market. So if anything, you would have to compare yourself to the other product with the other indication, or do a trial versus placebo to show the effect, or whatever. But that is, I think, a completely separate scientific advice that would have to be given, how to develop such a product for that indication. Yes, Andrea?
Maybe just to quickly add on this issue, and then we may need to stop because this is really a very complicated question. But I think that based on a comparability clinical study, we would not have the data on efficacy and safety to an extent as that we could accept this as the basis for a new indication. Normally, the biosimilarity exercise, also from the clinical part, is running over a few months, whereas in that particular indication, we have all in the back of our mind, we may want to see 1 or 2 years data, and I would doubt that such a comparison between reference and biosimilar would go to such an extent. But in the end, of course, that would be an interesting discussion for scientific advice.
Yes, please, there's another question.
Alexander Mensik from Amgen Europa Bio. I would like to change subject and ask a very general question to regulators, whether non-biotech products, which are biological medicinal products, and which may not necessarily be allowed to use the central authorization procedure, could be considered as biosimilars. Because you have those which are in the annex to the regulation, and those who are biological medicinal products, like a vaccine, for instance, but not necessarily biotech products. What are the views of the regulators as to whether or not you could have a biosimilar of a vaccine, for instance, and whether it would be required to use a central authorization procedure?
For a vaccine, I think it could be very difficult. It could be easier for recombinant products, but it could be difficult for other products. Very difficult, and much more difficult than doing a standalone development, I believe. So why would you go for that pathway if a standalone development might be easier? But the overarching guideline does not really exclude any biologicals, as you know, but we say that it is more likely that the biosimilar can be developed when it's well characterizable. So I think it's a high risk that you would take, and I wouldn't say it's impossible, but it's certainly a huge task. Yes, please.
My name is Michaela Flego-Kamensek. I'm from Baxter, and I would like to follow up on the BioBetter question. You mentioned that BioBetter has introduced intentional changes for efficacy and safety, but my question is, what is your opinion on changes in the administrative device, for example? Two ideas behind if the patent for the device is still protected, or if there is a development to increase patient convenience, what does this mean for biosimilars changes in that administrative device?
If you have a different device, that's a medicinal not a medicinal product if it's not directly connected. And that is, we realize that that such devices might differ. But that's wouldn't preclude biosimilarity if just the device is different. So please.
Judith MacDonald from Pfizer. I'd like to change the subject again and just return to the point, Dr. Weiss, you made about the tailored development, and that what is new in the current draft of the guidance is that there could be certain circumstances when clinical efficacy study may not be required, and the case could be made based on the PK and the comprehensive analytics and so on, and that is not always supported even by PD data. But it doesn't seem to be mentioned anywhere about, in those circumstances, what products would come into that category, and indeed, what about immunogenicity, and how would the immunogenicity aspect be covered in that situation?
Well, actually, it's a question I would have liked to pose to the industry, actually. Now, we are talking about an efficacy study may not always be needed. We are silent, actually, on the safety, and this is something really for discussion, whether or not in some cases where we have very well characterizable, usually peptides, polypeptides, non-glycosylated, and so on. Do we really always need a large, and we have, and do we really always need a clinical trial, including a safety trial or immunogenicity trial? That's really up for discussion, I guess. And my plan was to actually ask Dr. Emmert or Dr. Windisch on their views, whether they could consider scenarios, or maybe also the speakers from the innovator companies, of course, whether they could consider scenarios where this would not be needed, where we could be so sure that we wouldn't need safety studies. Yes, please, Dr. Emmert.
Okay, this is Karl-Heinz Emmert speaking. This is now my personal opinion, having worked in this area for quite a time, almost 20 years. So I think there is a lot of strange discussion about biologics. Biologics are drug substances than any other drug substance. They are manufactured by biological means, and their efficacy and safety depends on the physicochemical characteristics, on the functional assays, and in the former times they could not be well characterized. Right now they can be well characterized. We can establish similarity, we can investigate receptor binding, we can establish parallel and similar clearing mechanisms, and I wonder why we always assume there should be something strange with these molecules. Obviously, if receptor binding is the same, if physicochemical properties are the same, if functional assays demonstrate that it's the same, and when I mean same, I refer to laying within the variability of the reference product, why do we assume that there should be a difference in efficacy? So with regards to efficacy, I can imagine a lot of scenarios where an efficacy study is not needed. A different question is the immunogenicity. This is certainly a matter of concern, but there are molecules on the market for which it has been established that immunogenicity is not a real issue. And in such situation, I think it should be the responsibility of the applicant to demonstrate that it's justified that no additional immunogenicity study is necessary. I agree that in the present situation, in most cases, assessment of safety immunogenicity is needed. But again, let's see how science develops there and how we get experience in the future with these developments. But again, this is a personal comment from my side.
Yes, please.
Thanks, Richard Marquez with Amgen. And I think we all see the same way from an efficacy that there's predictive capability in functional assays, looking at the drug product, drug substance, and then we look at the drug product. And we have to keep in mind that we're doing a finite list of analytic comparisons and functional comparisons. So we're looking for specific aspects of the molecule, but some of these molecules are more complicated. So it's hard to think that you've actually characterized the whole molecule, particularly when we're looking at substance there, not product. And we have to conceive that the drug product's actually what's going to the patients, and there are no predictive models for how these analytic characterizations on a substance level can predict safety or, and more, like we're talking about, immunogenicity. So for a patient safety perspective, I think it's the right thing to do to make sure there is not increased safety concern or immunogenicity, since they're the ones receiving the drug product. So until we get real predictive capability, I think the clinical data is necessary.
So just to, you believe this is true for any biological, regardless how complicated or how well it can be characterized? Just to be sure.
It's hard to go to the absolutes, but in general, yes. It's hard to think of cases, like I said, where you can really convince yourself that you have predictive capability on a drug product level, including interactions with container closure, fill and finish aspects that did not have any alteration that could provoke some immunogenic response.
Okay, thank you. Jörg Windisch again.
Yeah, I always have to smile a little bit when we're trying to discuss such topics in general terms, when really they can only be discussed based on a data set that's actually been generated for a specific product. Now, 10 years ago, nobody would have thought that we can characterize glycan variants to the 0.01% level. We've made multiple steps, multiple orders of magnitude in sensitivity in the last few years. And then we're also talking about, and I agree with that, about simple molecules, the peptides, the insulins that could even be synthesized, some of them, if they're in the shorter period, could be chemical drugs. And then we're talking about different levels of complexity. But going forward, we will also look at the different levels of complexity in a different way because we're always learning more. So I just don't understand why we would shut the door on something when we have already made the experience over the last years and decades that science is progressing and that one day it may be possible. So today it may be possible for very simple molecules, but maybe in 10 years it will also be possible for very complex molecules. And I don't want to even that be understood as a general statement. Let's look at the data that companies put in front of the regulators and then we can make a decision.
Okay, I think Pekka Kurki would like to comment.
Pekka Kurki from the Finnish Medicines Agency and from the Biosimilar Working Party. I think that regulators always operate on a risk basis, so we evaluate the risks and try to concentrate our efforts where the major risks are anticipated. And here again, I think we should look at the history of the reference product. If we have seen a lot of problems with immunogenicity, I think we would be more cautious. If the patient population is somehow prone to develop immunological complications, lack of tolerance or something like that, we would probably ask for data. But there may be also cases where the history is very clean, and then we should consider whether we need to go and ask for further studies. Thank you.
Yes, Dr. Sumant.
Sumant Ramachandra from Hospira. I do support, and our company does support, a case-by-case basis based on the risk profile of the molecule. And it's not just the molecule, it's a drug substance, it's a drug product, and frankly, at times, it's a container system also, the primary container that the product is put in. As we have seen in some products, when you do change the primary container, you do make a change potentially in the safety profile of the particular product. So when when a company, whether it's the originator company or a biosimilar company, if it's a biologic drug, if there's a change in one of those systems, one has to think about what kind of safety data is needed. But I will say that you have to know, anytime you do a clinical study and you put a patient under a clinical trial, you have to know what you're looking for. So if it's a common event, you can cover that within a phase 3 clinical trial setting or even a phase 1 setting. If it's a rare event, you will need some sort of risk management plan in the post-approval setting to capture potential rare events. So it is on a case-by-case basis, and there is no one-size-fits-all. And it has to be considered, again, for the drug substance, the drug product, and the primary container that you're going into in assessing risk and post-market evaluation of that risk.
Yes, thank you.
Cornelia Ulm from Merck Serono. I can fully support what you just said, and I think here we can or we should profit on the product knowledge which has been gained over the years, because I think here in this particular case, if, for example, the originator makes a change or changes the container, then of course regulators evaluate whether that would require any additional risk mitigation activities, like doing and generating more immunogenicity data. And I think here we should really look as biosimilar products being biological products and put the appropriate measurements for controlling the safety of biological products in this view here.
Okay, thank you. I believe you had a question. Yes, back here.
I wonder, Josie Drapwell, IPOPI. I wonder if I may ask a question about adverse reactions. With the biosimilar products that are on the market, has there been an increase in adverse reactions? And if so, is that due to the active ingredient, the biologic, or a combination?
I guess this is a question to the regulators. And I'm not aware that there has been an increase. And nobody here, I think, from the other regulatory agencies is aware that there has been an increase in reporting or new adverse events, actually. Oh, lots of hands. I think you were first. Dr. Koech was first. Yes, please.
Sorry, Eugène Corrège, Sanofi. So back to the general question. Clearly, I think that nobody can disagree that analytical technology is progressing, and that what was not possible 10 years ago is now possible, and even more within 10 years. That's very clear. But I think that in parallel, there is also a discussion ongoing saying, basically questioning the meaning, the usefulness or the meaning of a clinical trial. So basically, the point is to say, I look at the innovator situation. I take one example. From this example, I make a general rule for different products, and then I extrapolate that to the biosimilar situation to be more specific. So we were hearing a lot that when you take two different products, two different innovator products, you don't see a difference in terms of efficacy in clinical trials. So meaning that basically all the products of the same class, in essence, in clinical trials, you cannot discriminate between them. And then the point is to say, if you cannot discriminate between innovators, then when you extrapolate and you go to the biosimilar field, what's the point of doing a clinical trial, in essence? And this, again, this discussion goes in parallel with the improvement of analytical technology. But the thing is that here we are comparing two different situations. When most of the cases, or in many cases, when innovator companies are making comparative trials, they are not necessarily looking for showing a difference. Maybe they want to compare themselves to the gold standard, and the objective is to show that they are equivalent, at least in terms of efficacy, and maybe having a better safety profile. That's one thing. And the studies, of course, they are not designed as biosimilarity studies. They are designed as non-inferiority studies or equivalent studies. And one important piece of the design is the dose which are used in these studies, because obviously the products are different, and to think that they could behave the same in terms of efficacy, you have to use different doses. That's very clear in most of the cases. And of course, and then saying that you see in these studies there are no differences, so there is no need to do clinical studies in biosimilarity, I think it's a bit extreme as a link, I would say, because again, different products that may be tested at the same dose, because they are different, they would behave differently. So my point here is to say, although we are not disagreeing with the fact that analytical technology is progressing, we should not throw away the concept and the utility of clinical trials in the context of biosimilarity.
Yes, I did not hear that anybody wanted to completely omit any clinical trials for biosimilars. That was not my understanding. I think my understanding was one should think about, in certain cases, whether it's necessary or not. I saw more hands, I think. Yes, please.
I was going to comment in terms of the adverse events, but let me comment again on the statement just made. This is Mark McCamish, Novartis Sandoz. So I don't think when we state some of the information that's been published regarding comparative clinical trials with different biologics, we're using that only to really attest to the sensitivity of the trial design. It's not a comment whether it should or should not be done, more or less from a sensitivity perspective that you can gain more information about the relative comparability of the product using some of the in vitro evaluations. And then some of these clinical trials can address many other questions such as immunogenicity that is going on. But back to the question about adverse events, all I wanted to say is we recently reviewed pharmacovigilance in our programs, and we have a fair market share in terms of biosimilars worldwide. We had well over 100 million patient day experiences with our products. And this question was really about naming and tracking and tracing. But as a result of looking at that, we did not see any adverse events that were unique to biosimilars in that comparability study or the evaluation of the pharmacovigilance data being said.
Yes, please. Dr. Suman?
Yeah, Sumant Ramachandra from Hospira. Actually, to a couple of points. One is to support Mark's contention. Actually, just I know this is not about naming, but just very quickly, Hospira just recently published a few weeks back also its experience in its pharmacovigilance database. We actually also have quite a large pharmacovigilance database, and we actually did not see that difference. So our data actually matches Sandoz's data, and we went in there and audited to make sure that we were seeing the right way. Just very quickly on safety and adverse event and picking up on clinical trials. In fact, I think because the originator industry has spent many years collecting a rich amount of adverse event data, and the regulators have it, when a biosimilar comes along, actually in development, we are looking to develop the product in a way that will avoid certain types of adverse events, such as immunogenicity, that could arise. And specifically the case I'm thinking about is in the case of erythropoietin, EPO, and where PRCA was seen with the originator. And over time, though it's not fully understood exactly what happened, we understood that there were potential 2 or 3 components that had to get under control, whether it was through the manufacturing process, the primary container, or through the shipping into the distribution channel. And the biosimilar companies, as well as the originator company, worked very hard to make sure today that we don't violate those parameters, so we avoid the occurrence of a fatal disease as a result of a beneficial treatment. So I would say the opposite. I would say that it's not about just new adverse events. It's a fact that you have to control and have the potential to control existing or known adverse event if you know the biological reason why that adverse event actually came about, if it's controllable. Not every adverse event is controllable. Some of it is due to the characteristic of the particular molecule or the route of administration, but sometimes it's due to the fact that it's the way it is either manufactured or packaged. And in the case of PRCA, we know that that is potentially controllable through the processes, and that's what biosimilar companies have done as well as the originator company.
Okay, thank you. I would just like to go back to the participants via Adobe, and there are some questions. First of all, there is a request that the speakers should speak clearly, because and again, identifying themselves. I think they're pretty good with this already. Again, here's Martina Weise speaking. And particularly in the light of the fact that there will be no written summary. So please be sure that you speak clearly. Then there is a question with regard to primary structure and post-translational modifications of the biosimilar, or if everything remains the same in the biosimilar and if there is still a better efficacy in the clinical trial, how do we judge this, and how does EMA treat such products? And would the sponsor have to revise the status and follow on biologics or biobetter? I think we have not had such a scenario, and personally I believe that if you're so close in your physical chemical characteristics and functional characteristics, the chance that you have better efficacy, show better efficacy in your clinical trial, I would say that's negligible, really. And if you still show really relevant better efficacy, something probably has gone wrong with that trial. Or there could be one scenario, which I think is discussed later today. If you have neutralizing antibodies and your biosimilar induces neutralizing antibodies to a lower extent, you could artificially create a situation where the efficacy of the biosimilar looks better than that of the reference product. However, that's not a real increase in efficacy, because you wouldn't really change the dose or anything in these patients. You would still have the same posology. However, you would over time have fewer patients with a loss of efficacy. And for such cases, and I think we come back to that scenario in the general guideline and non-clinical and clinical issues, we recommend that you should, if you know that from the reference product that that these neutralizing antibodies really do affect efficacy, you should have a pre-specified subgroup analysis for that reason. But other than that, I cannot really imagine why you should come up with a better efficacy. Martin, I think you were the next person.
Martijn van der Plas from the Dutch agency. Thank you, because you more or less said what I wanted to say, because I have struggling understanding the logic of these questions, and therefore I wanted to raise it, because either I do not understand it, or other people do not understand this. The point is that if there is a bit better efficacy, then there's something in the product or in the active substance that causes this better efficacy, and this difference can be detected. So if there is a better efficacy, there should be quality attributes who reflect this. So either the clinical trial is then problematic, or the CMC data are problematic, or there's something else that I currently do not think of that's problematic. And especially on the latter aspect, I would like to get some feedback if someone has any. Thank you.
Yes, please.
Keith Watson, Parexel Consulting. My feeling, if that scenario was to ever arise, would most likely be the assays are poor. In vitro assays are poorly sensitive and non-discriminatory, which, and I think that goes on to the point as well with, although I agree the quality profile should be within the ranges and should be very tight, we still can't underestimate there is inherent variability in any assay, and particularly biological assays. So I think the idea that you can be totally prescriptive on biological assays alone, I think too optimistic, in my opinion.
Yes, further comments on this?
I would like to come just back to the previous discussion what we had on the immunogenicity, or whether clinical trials would be in all cases required. I think here what we should think about is we are talking about a guidance document which should last for a couple of years. So I think here in a guidance document we should be open for any evolving possibilities. At the same time, we also have to think about the end users, like the physicians and the patients, and to educate those people about the biosimilars, because even if for small peptides or molecules where the regulators would not consider any clinical efficacy data required, that can afterwards for the marketing create really problems for acceptance. And I'm interested in seeing here whether the agency has any ideas on spreading out the scientific knowledge more intensive also in the medical environment to really make people understanding that if a product has been approved by the European Commission, that it underwent really stringent evaluations, and that really these inherent fears which are there because some studies have not been performed, or not really big efficacy trials have not been performed, that doesn't necessarily mean that the product is not usable.
You know that we all are communicating via, because we are invited to give talks, and we also have published, recent, it was in the last, not quite a year ago, in Blood, a manuscript exactly touching upon this perceived problem. And otherwise, if you have any other suggestions, we also now receiving input from learned societies on our guidelines, which is, I think, a very good thing. And we should, we also said we would be more proactive, because I think the learned societies is very important that we have them involved, because they also will certainly spread the understanding of what a biosimilar is. So those are our proposals. And I know that also EGA is doing a lot of communication and have handbooks and so on. So I don't know what we could do better, to be honest.
We all know that there are big conferences like for specific topics in oncology or rheumatology where a lot of clinicians go. I mean, maybe that would be something. I mean, I'm not involved in those organizations, but maybe that could be an idea to approach those conference organizers to really bring in some regulators to speak, to really enhance a little bit the force. Because, of course, I think that companies are already going into that direction, letting physicians who have participated in studies with biosimilars speak. But I think here it would be really good from a regulatory point of view to make really a better understanding about the rigorousness which is actually happening while approving those products, and really to make it clear that having approved a product in an ICH region versus a non-ICH region, that there is a true difference. And there's also one comment I want to make on the overarching guideline. There's one point where it said, I think it's in line 80 to 82, where they say the product is a close copy of a reference product. So I strongly recommend to avoid the use of the word copy in any way in a guideline, because that is really to differentiate between the non-comparable copies which are on the market in some countries, where we, from a quality point of view, clearly would not consider those products being compatible with biosimilar principles.
I think I saw that comment yesterday, and we will certainly think about this. Yes, well, we're taking your suggestions into account. Of course, I think we all have had lectures and talks also invited by learned societies. Myself, well, I have been invited to such occasions. It's difficult to be everywhere, you know, and I think this is probably still a slow process, but we had also referred to the EPARs in section 5.1, so physicians or people from learned societies can also inform themselves about how biosimilarity has been established, what the program looked like, and that it was not just a clinical trial, but a very thorough side-by-side comparison throughout the development program. So, but yes, thank you for that comment. I think we almost there, or we are there. Maybe we have a look whether there are any more comments from the Adobe participants. Do we have any more comments that we need to address? There was a question whether we could provide the link to the blood manuscript. Yes, we can do that. Yes, please. Last question in this round.
Thank you very much. Fabio Bisordi from Roche. And so my question is actually to the regulators on interchangeability. And the question is regarding if the EMEA should not be responsible for making also recommendations on interchangeability. This is actually a product property and not only a policy. This is a product property which allows the switching from a biosimilar to the reference product.
You mean interchangeability in the sense of the EU terminology? Not interchangeability as the FDA or it's interpreted.
Interchangeability in the terms of the patient and the physician making the decision to switch a product back and forth during the course of treatment.
I think that's not within the remit of the EMA, but I have to ask my EMA colleagues, because we just put in a wording in the overarching guideline that we are not making any recommendations on this. Can EMA people please verify this?
I mean, yes, we can very easily verify that it's not in our remit from the European Commission. So whether it should or shouldn't be, that's a different story, but it's not.
In this case, would it be possible to make it transparent that no additional data has been obtained on the switching of the products?
I mean, if such data are obtained, they will be discussed in the EPAR, in the European Public Assessment Report. So we will not neglect any data on that if they have been provided. Still, we won't make any recommendation based on that.
What about the recommendation within the SMPC that no data has been generated on switching?
Certainly not. Okay, I think we... Nana Kruse, please.
Nana Kruse from the Danish Medicine Agency. I just want to add that what you are searching for there is I mean, there's no difference from that a doctor can choose today to switch a patient from one originator product, insulin, to another originator product, insulin. That's free to do that, and nothing is stated anywhere in any SPC or anywhere else. Now we have a situation where we have one biologics which can be used for one treatment and another biologics. The biologics has been approved via a certain route of registration. It's approved as a biosimilar, but the doctor is still free to choose whatever product he likes. Of course, the situation is another if we go a step further and at the pharmacy level, they can pick from the shelf the cheapest product. That might require something further. But for the moment, if a doctor wants to switch a patient from one biologic product to another, he's free to do so.